Not only has the chemistry, physics, and biology changed since our first station, but the seas have kicked up too. We made it to our next process station, Station 4, where we will be for the next 4 days. One of the first operations completed was the deployment of the IPS system. Craig Taylor from the Woods Hole Oceanographic Institution will explain.
What I'm out here to do is measure rates of algal photosynthesis. And what photosynthesis is the use of light energy from the sun to fix carbon dioxide dissolved in the oceanic waters into algal biomass. They do this by oxidizing water to oxygen, which provides the reducing potential for the conversion of CO2 into organic matter. The way we measure the rates of photosynthesis out here in the ocean is to do what we call a tracer incubation study. In doing this study, we add a isotopic tracer form of carbon dioxide. In this case, we use the heavy isotope C13 carbon dioxide, and we measure the rate at which the algae incorporate this C13 carbon dioxide into their cellular tissues, and how fast they do that over time gives you the rate information that you're after. The way we're making these photosynthesis measurements out here in the ocean is by use of a new instrument that's been developed in my laboratory and that of McLean Research Laboratories in Falmouth, Massachusetts, for carrying out the manipulations of the tracer incubation study directly in the environment or in situ. The advantage of conducting these measurements in situ is that you accurately simulate the light field that exists in the environment, the temperature field, and the pressure field. So it gives a better chance of providing a more accurate measure of the parameter that you're interested in measuring. The way it's done is that the sample is drawn into an incubation chamber that acts very much like a syringe. So this syringe pulls the sample in from the environment and at the same time introduces our C13 bicarbonate tracer, and it automatically mixes with the sample and it incubates in that chamber for a specified period of time. At the end of the incubation, that sample is then expelled from the incubation chamber through a valve system to a filter that collects the particulate organic matter or the particulate algae that have now incorporated C13 bicarbonate into their cell tissues. Okay, the way we're going to deploy these instruments is on a free-floating array, float system. We are deploying 2 instruments at 2 different depths, and the array possesses GPS transponders that allow one to track the location of the array over time, and where it stays out in the ocean for 3 or 4 days. During each day, a primary production or a photosynthesis measurement is made repeatedly so that one can get a feel for the variability of that activity over time.
This array will be deployed 3 times during the cruise to sample the high, mid, and low productivity regions. Stay tuned for more updates.